Good day, this is Professor Will, CBMD PhD. Today's Sunday, April 11, 2021. It is 5:37 and 3 seconds a.m. Eastern Standard Time. Today's research, vaccinations. So let's hop to it here. New findings could lead to improved vaccinations against sexually transmitted infections. Date May 17, 2019. Source, King's College London. Summary. In a new study, the researchers show how skin vaccination can generate protective CD8 T cells that are recruited to the genital tissues and could be used as a vaccination strategy for sexually transmitted infections, STIs. One of the challenges in developing vaccines for STIs, such as HIV or herpes simplex virus, is understanding how to attract specialized immune cells, called CD8 T cells, to take up residence in the part of the body where the virus first enters. These cells need to be in place, armed and ready to provide an immediate protective immune defense, rather than waiting for immune cells in the blood to enter the tissues, which takes time. Before this study, it was thought that vaccines ideally needed to be delivered directly to the body's surface, e.g. female genital tissue, where the infection might start, so that the immune system can generate these CD8 T cells, travel back to the vaccination site, and eliminate any future virus that is encountered. However, delivering vaccines directly to the female genital tissue is neither patient-friendly nor efficient. Now the team from King's have found that their vaccination strategy marshals a platoon of immune cells called innate lymphoid cells, ILC1, and monocytes, in the genital tissues to work together and release chemicals chemokines to send out a call to the CD8 T cells, generated by the vaccines, to troop into the genital tissues. This research builds on the team's earlier work to develop skin vaccination techniques, using a dissolvable microneedle vaccine patch that once placed against the skin dissolves and releases the vaccine without requiring a hypodermic needle injection and generates immune responses. This study highlights how specialized groups of innate immune cells in distant tissues can be harnessed to attract protective CD8 T cells, arming the body's frontline tissues from infection. We now need to confirm these results with other types of vaccines from the one used in the study to see if a common pathway is triggered by skin vaccination. If proven, this could have a significant impact in improving the effectiveness of vaccines against sexually transmitted infections. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. I hope you enjoyed listening. Until next time, have a great day.